Good morning, everybody. Sorry, we're starting on a couple-minute delay. Thank you as well as usual for joining, and we'll give it another minute or so to let you all join in today. Uh, Hope everybody's having a good week. I want to first start off by saying we have a great presenter who's just a little delayed in joining us, but she'll be with us shortly. This week, we have upcoming, as many of you know, Juneteenth is coming up in a couple days, and we're happy to celebrate that. We do have, we'll send out a link, that the same link we sent before for our Zoom slides. We made a few Zoom slides you may see a few of our panelists using right now, Department of Medicine Juneteenth Zoom slides, as well as the Pride slides for this month. Feel free to, or backgrounds, feel free to use those, and they'll be reached at the link that will be sent out in just a minute. Dr. Dunn and Caceres did have a really nice diversity quarter message from our weekly newsletter, which we'll also send out in a link. And in that link, they talked about various things related to Pride Month, celebrating Pride Month to celebrating Juneteenth. They also note some events we have coming up, including one event next week during our Inclusion Diversity Round led by Dr. Terrence Mace, who's also been a ground one speaker with us. Many of you know him very well. And so we're really excited to promote those. So please shoot us a message anytime if you have any questions about the events that are going on, or events that you'd like to see happen, or things you'd like to see happen within the department. And I just want to make a shout out and a plug for really all of you that have helped make these Grand Round topics what they are. A lot of the topics that we do schedule come from, most of them come from feedback from faculty throughout the department. So thank you so much for the feedback. You'll notice that in the upcoming weeks, we're going to start expanding Grand Rounds. We're really excited to be expanding Grand Rounds to the point where we're not just doing COVID anymore. We're going to be covering all the topics we used to cover, very much in the same format. Throughout the year, we're going to be slowly migrating back to how we used to be, but we'll always keep the Zoom option. So for those of you who've gotten used to coming on Zoom, we will always have a Zoom option, but we are looking forward to also having a hybrid option, at least to start on some of our Grand Rounds, where people can also show up in person and maybe get some free breakfast. So with that being said, I do want to first have a couple updates. We don't have the usual updates today from Drs. Bowman and Dr. Pinsky. They're here with us, but number one, Dr. Pinsky mentioned that there's so little positivity rate. He actually doesn't have any variant data too much variant data to show right now. So that's kind of a good news. But I know that there is more variants that people have questions about, and Dr. Pinsky wants to update in the future upcoming Grand Rounds. And then secondly, Dr. Bowman is working on a lot of different things. And we just asked if we could hold the updates for next week, and he's going to have a lot of updates for us next week. We know that there's going to be a lot of questions with the California reopening. And so he and our staff and faculty are really working on making sure all those questions are answered. I want to turn over to Dr. Steph Harmon, who has some updates as well. I'm going to start sharing the slide here for you, Dr. Harmon.
Great. Thanks, Errol. I just wanted to share and announce the most recent Faculty Women's Forum awardees. So the Faculty Women's Forum is actually a university-wide organization really to promote equity and inclusion on campus for all faculty, and hosted and sponsored by the Office of Faculty Development for the whole university. And this year, out of the 16 awardees, 4 were faculty from our Department of Medicine. So I'm so excited to announce this and share this with you. They received their awards this past week, and there was a ceremony on Monday. First is Dr. Maya Rawson-Slater from our Division of Outcomes Research. She won one of the Outstanding Sponsor Awards. Dr. Joy Wu from our Division of Endocrinology and Dr. Sibel Renaud from our Division of Infectious Diseases both won awards in allyship. And Dr. Hannah Valentine and from the Division of Cardiovascular Medicine and our Director of Team Science Initiatives won one of the Deborah Rohde Lifetime Achievement Awards. Deborah Rohde actually is a law professor who passed away this past January, and had been basically the founder of legal ethics and the inclusion of justice law into our law school curricula. And so please join me in congratulating these awardees. Thanks, Errol.
Thank you, Dr. Harmon. And I also just want to join in congratulating our fellow colleagues. Congratulations on those wonderful awards. Just want to briefly mention, as always, next week we have Dr. Rahul Sharma, who will be talking about structural cardiology. And It's a special event for us. It's one of the first official non-COVID ground rounds we'll be having to really mark the fact that we're progressing through the pandemic. So Dr. Sharma has accepted that invitation, and he'll be presenting next week on that. We'll still continue to have updates as they relate to the pandemic and everything regarding that at the beginning of grand rounds as needed, including next week. The week after that, we have Dr. Glenda Gray, and then followed Dr. David Remlin, who will be talking about the question we'll be getting a lot of the COVID-19 origins as well. So thanks for those doctors. But today I'm excited to introduce Dr. Mark Davis. Dr. Mark Davis is the director of the Stanford Institute of Immunology, Transplantation, and Infection. He's the Burt and Marion Avery Family Professor of Microbiology and Immunology, and he's the Howard Hughes Medical Institute investigator. He got his BA from Johns Hopkins University, followed by his PhD from the California Institute of Technology. He did his postdoctoral fellow and was a staff fellow for the Laboratory of Immunology at NIH, and later become a faculty member for the Department of our Department of Microbiology and Immunology at Stanford, where he also served many, many roles. So something I don't have time to go over, but including the chair of the Department of Microbiology and Immunology. Dr. Davis is well known for identifying many of the T cell receptor genes, which are responsible for the ability of these cells to recognize a diverse repertoire of antigens. Other work in his laboratory pioneered studies of biochemistry, genetics, cell biology, and these molecules, including T lymphocytes generally, and how they play a role in orchestrating immune responses. His current research focuses on obtaining a systems-level understanding of the human immune system involving vaccine responses, including of the old and young, as well as connecting COVID-19 with autoimmunity and the problem patients have on immunosuppressive drugs. As you can imagine, Dr. Davis has a very active lab here that I think many of us are very well aware of. He's countless publications and awards that I certainly don't have time to go over now, but amazing history, a body of success and work. And we're just really lucky and happy to have him with us today. He is here today to talk to us about COVID-19 and the autoimmunity associated with it. I'm going to stop sharing my slides, Dr. Davis, so you can start sharing yours. Thanks so much for being with us today.
Great. Yeah, no, it's a pleasure to be here and to share some of the very recent work that we've been doing, and and I would just start out by saying that this pandemic reinforces the two things. One is that infectious diseases are still here and can have devastating effects. And also it illustrates how different people's immune systems are. I mean, we've seen this range in other things, but it's really stark with the numbers that we have in the pandemic, is how some people just never even notice they're infected, and other people become severely ill. So it's a great opportunity to learn about these differences in the immune system and what might be driving these different responses. Because I think in evolution, infectious diseases are everything. And there's a famous line in evolutionary biology that except in the light of evolution, nothing makes sense in biology. And I would say a corollary to that is nothing makes sense in immunology except in the light of infectious diseases, because there's a constant back and forth going on here. And what's revolutionary in terms of human diseases is this burst of technology that's been happening over the last 10 to 15 years, where suddenly, in the case of T cells, which is what we mostly are concerned with, though not exclusively, you can just do all sorts of things from single cells, from patients, with anything from anywhere, and then you can understand something about their T cell receptors. You can group millions of T cell receptors into subgroups that are sharing specificities, and then we could find out what those specificities are. And this has been one of the major activities of my group, but I think we're at a really good place, because we used to have to do this on a homemade level in terms of single cell, but now there are a whole bunch of commercial technologies that anyone can access. And at least part of this, not all of it, but part of it. And so there's several stories I want to tell you, but the most important is this one, and it goes back to some of our earlier efforts in clinical immunology and trying to understand particular diseases. And this was Arnold Hahn, which was a GI fellow, now on the faculty at Columbia, and he did this really interesting celiac study where he had a few celiac patients that on gluten-free diet who agreed to eat some bread, and then they get a mild response. And this also could not have happened without the collaboration of Nielsen, Fernandez, Becker, who we go to again, over and over again because of it's such the gut is such an accessible system. Anyway, long story short is that we saw something amazing with this gluten challenge, which is that not only was there a burst of gluten-specific CD4 cells in the blood at day 6, not unexpected, but what was unexpected was that there was also a burst of CD8 T cells and gamma delta T cells. And no one had ever seen this sort of constellation of mobilization. And so it was just an amazing mystery that we had to try to figure out. And really, it wasn't until 6 years later in a paper that came out 2 years ago almost, where Marisha Salagrama found the same thing basically in the EAE system, which is a mouse-induced autoimmunity demyelinating disease that actually Larry Steinman brought to Stanford. And we saw basically the same thing, that both
Davis,
in the blood.
Sorry to interrupt, Dr. Davis. Just
Oh,
make
sure.
We don't see slides being shared. Are you intending to share slides at this point?
Oh, sorry, I thought I was. Why am I not sharing a slide? Let me, yeah, wow, okay. Let's see. That's kind of critical. Okay, I guess I had started to, but then it had not. Okay, how's that?
Perfect, thank you.
Okay, that was what I was alluding to, this explosion of technology that allows us to just take from a blood sample to find out all kinds of things. There's a tremendous amount of information in the blood, not just white blood cells. There's a tremendous amount of information, and anything that's going on in a patient immunologically, you're going to see a reflection of that in the blood. And so there's a tremendous amount of information that we can gather. It's been part of, deciphering what you see is of course a critical thing. So anyway, what I was saying is that in celiac, in a celiac challenge model, we saw this mobilization of CD4, CD8, and gamma delta T cells, and then it goes away in the blood. And so then we went to this EAE system, and we saw the same thing. That 10 days after the induction of the disease in these mice, we saw mobilization of CD4, CD8, and gamma delta cells. And that was in the blood. Being mice, we could look in the nervous system, and we saw exactly the same thing. And then a week later, there was another surge of the same cell types. And to some extent, you can sort of see a third surge there. So there was a really interesting periodicity to this, suggesting opposing forces of some sort, maybe. And the EAE is not supposed to be a model of relapsing remitting MS, but here we are seeing a periodicity that seems to resemble the kind of back and forth that you see in many autoimmune diseases. Is that the disease is quiescent for a while, then there's a flare, and then it goes down, and so forth, and that really encouraged us to think we were onto something important, and also going back and forth between the mouse models and humans to see this convergence. Sorry about the dog. So then the mystery was that we could address much more easily in mice than in humans is, what are these things doing? What do they see? And then that was fairly straightforward for the CD4 cells. They were seeing the MOG antigen that was used to induce the disease. And the gamma delta cells were turning into IL-17 producers, which is also known to be part of the pathogenesis of EAE. But the mystery then focused on the CD8 T cells, because they didn't see MOG, they didn't see anything. These are, we take T cell receptors and we implant them into T cell receptor minus T cells and then apply them with different antigens. And bottom line, we looked at a ton of myelin antigens that have been nominated to be important in MS or in EAE disease, and we got nothing. Nothing was happening there. But luckily, again, part of this wave of technology, Chris Garcia had been developing this a fantastic yeast display system whereby you could take a given T cell receptor and screen a yeast library that was displaying 1 billion different peptides in a given MHC molecule. And on a good day, you can then pull out the peptides that might be important. And so we did, and we got 2 peptides, and we thought, okay, CD8 cells, they're gonna, they're all, we know that CD4 and CD8 cells work together to suppress viruses, for example. Is that what's happening here? And so if we induce the CD8 cells specifically, we will be enhancing the disease. Well, of course, the opposite happened, which is that we suppress the disease, that normally you get a good clinical response in these mice in every mouse. But when we put in these CD8 peptides, it suppressed the disease. Most of the mice didn't get sick. The few mice that did get sick, there were like 3 here out of 10 had a very mild disease. So we're looking at a suppression phenomena, and that explains now the yin-yang aspect, the periodicity, because you can imagine there's a surge of pathogenic cells that then is counteracted by a surge of suppressive cells. And it turns out that this represents the cells that do this are not just all the CD8 cells, but they're a very particular subset of CD8 cells, which display this marker Li49, or a family of markers, which are inhibitory receptors, characteristic of natural killer cells, but they are on a subset of about 5% of mouse CD8 cells. And it was about killing. So basically these cells could kill. And in fact, the normal CD8 cells could not kill. These are MOG-specific CD4 cells here being killed or not being killed by the most of the CD8 cells, but they could be killed by this Li49. So it was a small subset of CD8 cells that is counteracting this pathogenic response. And so that was incredibly interesting. And we're really anxious to get into humans and to look at human autoimmune disease. And this has been the work of a fantastic postdoctoral fellow, Jing Li, for the most part, who's really driven this project tremendously. So Li49, the equivalent of Li49 in humans is the KIR family of inhibitory receptors. Again, they're on they're characteristic of natural killer cells, but they are expressed on about 1 to 2% of healthy controls, as shown here. But then with help of PJ Utz and Bill Robinson and others, we were able to get samples from lupus patients and from MS patients from MS clinic here. And we saw in a few cases, amazing levels of these KIR positives. So something was going on such that in some cases you had 10 times the normal level of KIR positive CD8 cells. And the general run of the thing here is shown. And even more dramatic, again, with the Nielsen, Fernandez, Becker, we looked at gut samples, and these, you see even more of these cells, about 5% in healthy people in a gut biopsy. But in celiac patients in remission, you see a higher level typically. And if you're just having an acute episode of celiac, you get these even large, higher numbers. And we also are able, in celiac, because we can analyze the pathogenic gluten-specific CD4 cells, we can measure the number of these cells versus the KIR positive cells. And there's a linear relationship, again, suggesting there's a yin-yang thing, that the more pathogenic cells you have, the more you need these inhibitory cells. And then Jing did this really brilliant experiment where she was able to take blood cells from celiac, separate the CD8 cells away from everything else, stimulate the everything else, which includes CD4 cells with gluten, raise up the gluten-specific cells using a reagent from Ludwig Solid. And if you add KIR-negative CD8 cells from the same patient after stimulation, basically nothing happens. But if you add KIR-positive CD8 cells, you wipe these cells out. That 90% are now killed, and in most cases. So this says they're not only phenotypically equivalent, but they're also functionally equivalent, and that in actual humans and actual patients, we can see this effect very clearly. So then things got even more interesting because we were seeing these cells, and we did a lot of transcriptional analysis, and they basically were very similar between healthy controls and people with these immune diseases. And so it became clear this is really a normal mechanism. You don't have these cells just to do something about autoimmunity, that they're a normal part of your working immune system. And so that's when you start trying to think broadly, and it connected very nicely with this older work we had done, published in 2015. And this is Wang, who was at the time was a hematology fellow at Stanford. What we found was that despite 25 years ago, there was very clear, or 30 years ago, actually, there was very clear mouse data that said that if you're a self-reactive T cell and you see that antigen in the thymus, you're dead. That you basically wipe out all the self-reactive T cells you can find in the thymus. And then that was called central tolerance. That was supposed to be the main mechanism of tolerance and preventing autoimmunity. But as I said earlier, autoimmunity is not a big deal evolutionarily. If something that negatively affects less than 1% of children and young adults doesn't really matter evolutionarily. What does matter is infectious diseases, where historically for thousands of years, half of children essentially died of infectious diseases in childhood. So people had larger families because that was kind of the rule. And it was only vaccines that have shifted that picture, so that now very few children die at those ages that they used to die in droves. So what we found, though, just looking at healthy people, samples from the blood bank, which is a great resource for looking at healthy immunology, there were lots of self-reactive cells in healthy people. That the mouse data was actually an artifact of some of the transgenics and other ways we look at it. Was the only way to look at the time. Now we have more sophisticated methods, and we find lots of self-reactive. There is some negative selection. There is some deletion of self-reactive cells, but mostly everything's there to some extent. And to try to explain this, we proposed that this was because of infectious diseases, you didn't want to delete anything from the repertoire. You might get rid of the most dangerous cells, but in general, you keep everything around just in case this particular mutation comes that fits, which is going to have the logic of a lot of immunology anyway. You create enormous, you can't possibly evolve faster than a bug. It's no, not even close. 30-minute replication time versus 20 years. There's no contest there. You can't evolve, you can't out-evolve a pathogen. But what you can do is create massive diversity such that anything that comes along is likely to find a receptor, to find an antibody, to find a T cell that's specific for it. And so these cells are sitting around waiting for their own special infectious disease. And also the other interesting thing is that classical work in mice has shown that, at least in a number of cases, infection breaks tolerance, that normally you control these cells, and we have evidence about how they're controlled. But when an infection happens, you might die, you're getting, you're unleashing the Kraken, you might even say, and you activate all these cells, some of which will be self-specific as well by our thinking. But then if you do that, how do you control them? You now have unleashed dangerous cells, and they need to be controlled. And we thought, well, maybe that's what these positive cells do normally. And that it turned out, then you need people with an active infection. And as you may remember, there was this massive pandemic, and hospital was filling up with people with COVID. And so we looked at that, and that was tremendously enabled by Kari Nadeau and her clinical team that just provided us with massive numbers of samples, blood samples from COVID patients at all different stages. And so we were able to do this experiment here, where we could just see, in fact, that instead of the 1 or 2% normal level of positive CD8 cells, even in mild COVID patients, they're going up, and in moderate, they're more so, and severe, really very high up. And yet, you might also ask the main thing people have focused on in terms of tolerance in periphery have been CD4 Tregs, and they're clearly very important for at least some aspects of tolerance, particularly fetal tolerance and so forth. But bottom line, they weren't doing anything in COVID, but these cells were. Also, we could see that with autoimmunity, particularly with the vasculitis, which it's not really clear how close that is to classic autoimmune vasculitis, but they were doing the worst. And yet again, CD4 Tregs weren't involved in this. But it also fits with mouse data, where if you delete CD4 Tregs, they have no obvious effect on acute infection. We did also get a few people with acute influenza infection, and we could also see that they also had elevations in the seropositive CD8 cells. And again, CD4 Tregs were not doing anything. So we think these cells are it in terms of having this very specific role in infection and suppressing. So we think every time you get an infection with something, you will be activating self-specific, potentially dangerous T cells, and you will be activating through some feedback mechanism, you'll be activating these seropositive CD8 cells. But in autoimmunity, something goes wrong with the circuitry. But it's also worth remembering that people with autoimmunity, they're not born with autoimmunity typically. They are 20, 30, 40, 50, 60 years old. They've had many infections, but something happens such that you usually can link the infection with onset of autoimmunity. That's a well-known phenomenon. And the explanation for it is quite reasonable, is that there's some cross-reactivity between the bug. But it hasn't been thought that there's a broader mechanism, and especially a mechanism involving yet another. Some of you might think there are already enough subsets of T cells, but this is what's happening here, and here is another subset, and it's clearly very important. The other thing we've done, Jing has done a lot of sequencing of T cell receptors. We're kind of fond of T cell receptor sequencing, and we can see similarities in critical bits of TCRs called CDR3 regions that are the bits that bind the peptides. And we can see commonalities, so between celiac and healthies, COVID-19, in this case, type 1 diabetes, COVID-19, lupus, more lupus, celiac, type 1 diabetes. So it's clearly it's a broad response, but there's a lot of commonality, and that is interesting in the sense that maybe a drug targeting one of these T cell receptors or more could be more broadly applicable in terms of these other diseases. So the story here is that this represents a new and I think very general mechanism of peripheral tolerance, and that there's a general thing that you need to suppress dangerous T cells that will come up in an infection, and yet you want to survive the infection, so you have to give them some scope to do something. But you've got this whole mechanism that is like a hammer that will come down and help you out. And this is Jing, who's just about to stain these CD4 COVID-19 samples. And then I got a few other tidbits to tell you about, particularly the questions about COVID-19 directly that we've been exploring. And one of the main mysteries is why, or if some people have no symptoms, and others become very sick. And there's been this thought that pre-existing immunity to circulating coronaviruses might be important. I think we can confirm that, and we can do it by, so Mamsi Veldosa has developed a new form of multimer, a peptide MHC multimer for staining T cells. Now, the history of this goes way back, because 25 years ago, John Altman and I developed these tetramers, which are basically, you put a biotin on the end of a MHC peptide complex and complex it to streptavidin, you automatically have 4 peptide MHCs. And that's been a very useful reagent for years and years in both basic immunology and clinical immunology. So Vamsi thought he could do better, and particularly by taking advantage of ferritin, which is a 24 subunit self-assembling nanoparticle, essentially. And so what he did was put a biotinylation signal at the N-terminus of this ferritin, and it turned out expressed really well. And he's able to bind peptide MHCs, 12 peptide MHCs onto this thing, and very easily, and using a lot of existing reagents. And it just works really well. And we call them Spermers. And here's some of the staining data. The original tetramer, we always knew the original tetramer, always missed some cells. And you can see that staining here in a mock-up. There is another kind of multimer called Dextromers, which is a polymer of dextran that decorated with biotin. And you can bind a lot of peptide MHCs, streptavidin peptide MHCs, but it's messy. It's a polymer. And so it's not, you have a distribution of molecules and not a uniform molecule. And that's what I think where the Spermer shines, is that it's a very uniform molecule. It's self-assembling. It's runs very nicely on a sizing gradient. But the other thing that really was surprising, we didn't expect to see this, but it actually collects a lot more T cells than normal. So this is a flu reagent, for example, and we're getting 3 to 4 times more T cells being collected. A little less, well, that's another story. These are tetramers, so you only get what you get. But here, this is more relevant. So this is a flu antigen, this is a CV antigen. We're reproducibly getting 3 or more times. So if you're in the specific T cell business, this is a really nice thing, and more sensitive, and you have more things to look at, and so forth and so on. But in terms of the question of does pre-existing immunity to circulating coronaviruses matter in COVID? And so Vamsi did a big analysis of the 4 circulating coronaviruses before SARS-CoV-2 and SARS-CoV-2, and identified a whole bunch of more or less conserved epitopes, peptides, which is what the T cells see. Most are not in, I was supposed to get there yet. Okay, all right. So then he took 24 of these and made Spermer reagents and looked at first healthy controls, unexposed people. And what was really nice to see is that in low conservation, that is peptides that were unique to SARS-CoV-2, you mainly stain naive cells in naive people, all right? But in the same subjects, if you look at a highly conserved peptide, you see memory cells. These are 2 different flavors of memory cells, very few naive cells. So that says that there's memory cells are characteristic of responses that you've already made. And so this is really good evidence. And of course, there's a lot more of it, that there is some pre-existing immunity. And memory cells are what you want. Memory cells are by far the most active in terms of infectious disease defense. That's what you want memory cells. However you can get them, you want them. It's the difference between never even noticing you have a disease and dying from it. So it's a big deal. That's what vaccines are supposed to do. That's why you get boosts to boost up not only the numbers of specific cells for a given pathogen, but also to push them into this memory phenotype, because we know that that's a much more potent type of protection. So, but does this matter in terms of COVID? Well, again, working with Kari, it's been tremendous in terms of just having a lot of samples to look at. And sure enough, patients with mild disease, purple here, are recognizing these conserved peptides, or making a robust T cell response, CD8 T cell response to these conserved ones. People that have severe disease seem to be favoring these unique peptides, maybe because their memory cells are not up to speed, or I think it's also, because of the increased mortality in older people. We know that older people lose immunity, something called immunosenescence that we've studied, and others as well, is a big deal in terms of people that 70, 80, 90. Their immune system starts to lose functionality, and partly that loss is in memory cells. So anyway, this is strong evidence that pre-existing immunity, T cell immunity to these conserved parts, bits between the different coronaviruses matters clinically. And in fact, maybe one of the only evidences of T cell correlates of protection in this disease. And I should just say this is MC and doing great stuff. So now we get to the original title here. I hope you haven't had to wait too long, which is this quite large problem about immunosuppressive drugs. And I think many of you know this, that billions of people are on immune modulatory drugs, organ transplant patients, patients with different kinds of autoimmunity. A lot of people are on these drugs, and in many cases, they're not making good vaccine responses. So they can get the full round of vaccines and yet not have antibodies that we know are extremely useful and helpful for putting things off. So this runs the gamut of this is just talking about COVID-19 patients treated with infliximab because of IBD and not making great responses. Bottom line is that, so this is probably the important thing is that these are the infliximab patients, and these are the anti-SARS-CoV-2 antibodies. And this is a control antibody that doesn't depress immune function, but does something else. I forget what. And you can see this is a sort of normal response. This is a patient response. So you can clearly see this not doing well. And then there's papers, particularly this one by Yarsky at Johns Hopkins, and they analyzed transplant patients. And transplant patients are probably doing the worst because you get very serious immunosuppression to repress graft rejection. And it affects, it's aimed at T cells. So anyway, they did this big study, and clearly there's a problem. This is just some of that data. And there's other data. This is a group in France. They're one of the few people that have done a T cell response in kidney transplant recipients. And again, not good. Not good antibodies, not good T cell responses. And then there's some other data, Washington University, glucocorticoids. So we have all these, and a whole medicine chest of things that modulate the immune system, mostly downward because you've got an overactive immune system, or are you trying to reject your organ or something? And yeah, so there's a lot, and so now, of course, there also have been studies at Stanford, and we've been involved in some of them. Hannah Valentine, Carrie, Scott, PJ, myself. It'll be interesting, especially on the T cell side, to be looking at this, and also just to run people through kind of a whole course. When you're doing a serious vaccination study, you really need multiple time points to see what's going on. And so again, I mean, we know bad things are happening, but sometimes some people are making responses. What are they doing right? What are other people doing wrong? What are the nuts and bolts of these poor responses? Are there, and especially in the autoimmune case, we really don't know much about the T cell response, and that might save you, and I'll mention that a bit. Right, so Lisa Zaba and PJ Out-Sankari are doing a nice rigorous study of vaccination in lupus, psoriasis, and tumor immunotherapy patients. Kari organized a really nice 55% healthy control cohort, which I'm one, and that's really going to give us a good baseline for what a normal response is like. And then just luckily, Stefan Busk is able to give you these slides where he's done a really very comprehensive, with Eileen Wang, a very comprehensive analysis of antibody responses in kidney transplant recipients. And the news is terrible, basically, that they're not, these are healthy controls or pre-transplant, and these are all the transplant patients. So it's pretty terrible. And these people are at risk. You may be fully vaccinated, but you're totally at risk for infection if the antibody is critical. And again, there is some there's some evidence of that. Yeah, I don't know. And Ron Levius also has a big study with Ashwalazade and Panya Shri in patients with lymphoma. Patients with lymphoma are routinely given rituximab, which wipes out your B cells. And you might imagine that wiping out your B cells is not a good idea if you want to make a good B cell response. And indeed, I think there's already some evidence of that. Actually, previously, there's even a paper, I think 10 years ago, I saw where sorry about that. Yeah, so, but these are the studies we need to do, because we need to really look at what's happening, what's going right, what's going wrong. And also alert the patients. I mean, I think that word has gone out to a large extent, but if anyone has patients on immunosuppressive drugs and hasn't told them, this is a big deal and they need to be informed. And the other good thing is, though, that the pathology department, courtesy of Scott Boyd, developed the state-of-the-art antibody testing, particularly a mock-up of what's called neutralization assays, which is the critical thing. The most important thing in preventing these kinds of diseases is to inhibit the virus from getting into cells. Once they get into cells, you're at another stage to worry about. But if you can prevent that infection, which the very best antibodies do, you're in good shape. And that's really part of also where the answer is going to come from. I think for organ transplant patients particularly, they're not making good T cell responses, they're not making good B cell responses. They're in serious trouble. But that's where passive antibodies, a number of companies, including one I've been associated with, Beer, but also Regeneron particularly, have developed monoclonal antibodies that do just that, prevent the virus from getting into the cells. But there also might be other strategies. Because there are multiple, we have these multiple vaccine modalities, not just RNA, but also adenovirus and other strategies. These are different, very fundamentally different types of vaccines. And so it's possible that If one fails, the others might work. But I would say for people with the severe, if you've depleted your B cells or you're on anti-rejection therapies, then probably passive antibodies is going to be the thing. But there is hope in terms of T cells. And that's one thing we're doing a lot of, is using this Spermer technology to look at T cell responses in vaccination. And there's hope there because there's this paper in monkeys. So you can only do this experiment in monkeys. This is Dan Baruch at Harvard, Mass General. And basically the bottom line of this study, and monkey disease is quite similar to humans, what you really want are the high affinity neutralizing antibodies, all right? But if you don't have that, you really want T cells. And he basically was able to show this in this paper by depleting T cells in the monkeys, and then things got bad. So we have more and more evidence that the T cell response could be very important. It could save you. We don't know that, but there's that possibility. And this is another paper about T cells where they, Alexetti La Jolla, basically was able to show that you have massive T cell response. There are lots of things that T cells are recognizing, and they also don't care about the variants. So of course the variants are a big problem now because some of the antibodies don't work when you change the spike protein, which is what viruses do. They change whatever it is the immune system is going after, they can mutate and change it. But it's so far, at least in terms of what Alex has published, the variants are, he's not seeing an effect on the T cell response. And then this is a really most interesting data that Sean Tucker pointed out to me, which is that there was this very small study of people with antibody deficiencies, and including XLA, which is basically X-linked immune deficiency, basically no B cells, and yet they recovered. They didn't go to the ICU. And then there's common variable immune deficiencies, which also deficient antibodies. And there was some not so good, others recovered, and not necessarily ICU. So this is some evidence. I mean, I think in monkey evidence may be more concrete, but there's at least some evidence people with immune deficiency and antibody deficiencies might actually be able to get by just on the T cell response. So that's a hope, and thanks for your attention. I'm happy to answer any questions here, and particularly though it takes a village to do these things. I think we have this mythology in academics of the lone hero going into the cave and slaying the dragon, and maybe that worked in the Middle Ages, but really for human work, you really need a team. You really need clinical expertise, science expertise, technology expertise, computational expertise. I mean, it's a party. But I think we've got a really good group of people here that we've worked together for years, and I'm very proud of how well people have done in the COVID business. But in my lab, I pointed out Vamsi Malajosala, Jing Li, earlier work by Marisha Salagrama, and many others have contributed here. So thanks for attention.
Dr. Davis, thank you for that presentation. I'm going to get right into just already a lot of questions. I'm going to try to get through as many as we can before we hit the hour. One question we've been getting a lot all year, and I'm looking forward to hearing your thoughts on this. This question, so I have a couple questions that are very similar. One asks, how do you judge the success of vaccine in patients? And then Dr. Louis is asking more specifically for immunosuppressed conditions. How many patients? There are many patients asking for the antibody test, and many of those patients are immunosuppressed. Should we be ordering these tests both for immunosuppressed patients, all patients, and how do we interpret it if it's quote unquote low? What does that mean, and so on?
Yeah, I think anyone on immunotherapies should get tested, and the pathology department has a great CLIA-approved set of tests, state of the art. So that's really worth knowing. And if you've got good antibodies, you can, rush out to the nearest bar or whatever you want. I keep asking whether there's still discos, but anyway, that's but I think the mystery though is the T cell response. To what extent, if you have a good T cell response, to what extent is that going to help? Is that also a get out of jail free card? And we just don't know. I think that's been the problem. The T cells responses are very complicated. I've spent a lot of years working on that. They're very complicated, and we still don't have T cell correlates of protection for any vaccine, just because of that. I think we're close to getting somewhere, but you can't tell a patient, hey, you've got a great T cell response, you're free to go. No, we don't know that. But I do think that the evidence of the passive antibodies that people have been developing, and that some they're starting to say you could just get an intradermal injection of an antibody, and that will last for months. That's probably the best bet at this point. I think right now you can't just get that for asking you, but if you have an infection, if you have a virus infection that's positive, you should immediately get those antibodies. No question about that.
Great. Thanks so much. I'm also, Dr. Pinsky, feel free to comment. Thanks for messaging the panelists and attendees here. And many of you know Dr. Pinsky has been answering specific questions to the actual tests we have and how they're used and so on. So feel free to comment. There's another question by Jessica asking something similar, which antibody test. So I think Dr. Pinsky is answering that. I'll jump over to the next question. Dr. Barman had asked earlier, do we see different rates of long COVID in patients on immunosuppressant drugs?
Yeah, that's a really interesting question. I don't think we know the answer, I think. But I could imagine, the real danger is people not clearing the virus. So if you're seriously immune suppressed, you're not going to be clearing the virus. So you're going to be lingering on, and you're going to be a carrier. You're going to be a spreader. And there are already cases of that. Months and months after initial infection, people still having virus that they can share with other people. That's and identifying, and those are going to be people with either natural immune deficiency or probably on these drugs. So I think that's another aspect of it. They should be continually monitored with, because they could be a danger to the community.
Dr. Kim asked, thinking about this in context of transplant tolerance, I would like to ask whether KIR positive and CD8 positive cell functions remain HLA restricted, and whether there is a path to induce donor specific tolerance.
Yeah, that would be a dream, wouldn't it? They are specific. We do know that the T cell receptors are working away, and that's why we see the sequence sharing, specificity sharing. So that's definitely a factor. Right, could you? Well, one obvious thing is to ramp them up, and that's the mission of a company I've helped to found. Can you figure out some way in which you can push these cells higher to jump higher and do more of what they're doing, assuming that it's all working, and just overwhelm somehow. So yeah, that would be the dream, is that by manipulating these cells, you could clamp down on graft rejection or something, autoimmunity.
I have a question here from Dr. Chu. He asks, in recognition of conserved peptide correlates with mild COVID-19 disease, could there be improvements to the current vaccine strategies?
Yeah, absolutely. And that's what I've been telling anyone who listens, that don't just, the spike protein actually does not have a lot of conserved epitopes. It's the other proteins in the virus, like the one we looked at, the open reading frame one I think it's called, that had lots of conservations. And so I think a good next generation of vaccine would include that protein. And that, I'm sure that would help.
Great, thank you. Next question here. I'm going to go to Dr. Leibowitz's question, and Dr. Schleicherman. Dr. Pinsey is going to answer your question about the specific antibody test that we have in the lab. Dr. Leibowitz asks, a question about immunosenescence. Are those who have it more affected by who have been more affected by reduced response to disease for which they have previously had immunity, for example, polio, mumps, measles, chickenpox, or is a disability to respond to new diseases they haven't had or been vaccinated against, or both? I haven't heard of immunized older people getting measles, mumps, or polio, but they get shingles. What's happening?
Right. Yeah, that's what I think one of the problems in the vaccinology is we see all kinds of variation in how long immunity lasts. So the yellow fever vaccine, too bad yellow fever isn't around much anymore, but if it is, that vaccine lasts 60 years. People, you can assay people 60 years after they got it, and they're still happening. But then other ones are fading. And I think that shingles phenomena is basically one of multiple cases of fading immunity, and where you need a boost can help. At least now in shingles, the latest vaccine, which I think is called Shingrix or something, that actually is like 90% effective in preventing shingles. So there's some good options there.
There's a question here. Are there studies on how vaccine interacts with drugs like Entyvio? Do such drugs affect the immune response?
Yeah, I mean, this is one of the interesting things about modern medicine is that no drugs are tested for immune toxicity. They're tested for things that are likely to work in, and then if they work, people say, that's great. But I think what we're seeing here is chickens coming home to roost because we don't know very much about how those drugs affect immunity in general. And we recently developed this tonsil organoid system where we can basically make tonsils into working lymph nodes and vaccinate them. And so testing drugs for toxicity, this will now be I think a really good test bed to test drugs for inhibition, for immune toxicity, basically in terms of inhibiting a vaccine response. So that should be very good.
Great. Are T cell responses equally potent against different variants?
No. There's something called immune dominance, which is somewhat exaggerated. I don't think it's as important as people have thought from mouse work. But definitely you get more of some specificities than others. And no one quite knows what that is. I think there's a simple explanation, which is that some peptide MHCs have more potential solutions in terms of antibodies and T cells than others. And so the ones that have more solutions are going to be more popular. You're going to see more T cells and more B cells with those specificities, and the other ones are you're going to have maybe a much narrower window of specificity to work with.
Dr. Davis, I might end on this one last question, and it's from Dr. Valentine. It's a great question. There's data suggesting 3 doses of the vaccine might be helpful in transplantation in patients. Is this likely and worth studying?
Well, definitely worth studying, and we should try everything that's even remotely possible. But I do think that when you're suppressing graft rejection, you're really putting an anvil on top of the immune system, both the T cell part and the B cell part. So I'm not incredibly hopeful that will be the answer. I think it's going to be more about passive antibodies, which luckily there are. Luckily people have been developing them in they'll be available in it. And I think that's probably going to be the solution.
Well, Dr. Davis, I think we'll go ahead and close it now. I just want to thank you for all the work that you are doing and have done. And thank you for being with us to present today. Again, as always, thanks to the panelists for being with us and for all of you for hanging with us today. I hope everybody has a great rest of the week, Juneteenth as well, and we'll see you next week. Thanks so much, Dr. Davis.
Okay. Thanks, Errol. Thanks, everybody.
Bye-bye.